All right, Mark, welcome to the show.
Thank you very much for asking me on.
Yeah, no, it's great to have you here. Honestly, I'm really excited about this conversation after Andrew mentioned, like, he sort of gave me a sneak peek of the story, and I was like, oh my God, we got to do the show. Like, we got to have this conversation. And so, Mark, just for everybody that's listening, sort of, can you give us a little bit of your background, because you're not the computer science born and bred person, right? That a lot of the people that I will have on the show, you're hardcore drug discovery.
Yeah, I mean, I had one programming course in college that I actually took to fulfill a foreign language requirement, because programming languages were the neat new thing, and they wanted to encourage that. So yeah, I have no sort of technical knowledge in programming, and my background, my training is in clinical pharmacology, and basically spent the last 30 years in the drug industry, in big pharma and CROs, on non-clinical, preclinical development and clinical pharmacology, PK/PD. And before I started twoXAR, I was consulting for a while, and before that I was at Jazz for 15 years, or not 15, it feels like 15, like 12 years, starting out as VP of research, and then had various titles along the way. And along the way I became inventor on something like 30 patents, including the original ones for the antihistamine Allegra, and then at Jazz on some of their Xyrem patents, and just had, spent my time in developing drugs. That was my passion and what I like to do, especially the early stages of drug development. And then started consulting after I left Jazz, and twoXAR was one of my first clients. So that's how I got to them.
So essentially, what I would consider, a traditional drug development background. Well, I mean, obviously distinguished, like you've done some incredible stuff, because my son takes Allegra, so I'm sure he's very happy that you developed it. But, I guess your first exposure to this was dealing with twoXAR as a client, if I understand correctly.
Yeah, that's right. So, they were mostly computer and bioinformatics people. They had a few people with drug development background, but they realized they needed more in that area. And through a friend of a friend, they reached out to me, and I was consulting. And so I said, sure, I'll be happy to help them. And, what I knew about AI at that time was like from documentaries I had seen about, computers beating chess. And then there was a one,
Right.
The more recent one about AlphaGo and about the
Yes.
Go masters, and
Yes.
the team from that, Google bought them, and they're playing the master, and the computer program wins, and they're oohing and aahing about this, and they can't figure out why the computer, made this winning move, or why it came up from that. And, that was my understanding of AI. And I couldn't understand how AI could possibly help with drug development, because I could see, okay, for a game, I can see how a computer could play against itself over and over again and figure out what are the winning moves, and learn to think ahead and things like that. But for drug development, if you use that game analogy, and if you use like success as an FDA approval, then there's only a few thousand games that have ever been played. And so how is a computer supposed to iterate on that? And if you define an approved drug as a win, as the target that you're going after, then how are you ever going to come up with a better drug, or a drug for a disease where there aren't drugs approved, which is the goal. So I didn't understand how AI could help with that. But as I became consulting with them, they obviously didn't bring me in for the AI part. They brought me in to say, okay, we found this, we found this interesting molecule and we have these, how should we test it? Or we've tested it and now we have these results. Help us
Right.
interpret that, or help us with the next step. And, I would do that and I would say, oh, this looks pretty good. This is neat. I wouldn't have thought that might happen, but obviously it did. And in sort of retrospect, they can figure it out. Okay, so now we should do this, and I would see that, that pattern would repeat itself where they would run their computer platform and come up with like 9 or 10 molecules and put them in a non-clinical model of efficacy, and 2 or 3 of them would pop out positive. And the hook for me was they all had new mechanisms of action. So these were mechanisms of action that hadn't been tested in the patient population for that disease. And so, that was an immediate challenge to my understanding of AI. So how did a computer come up with that? And I didn't know the answer to that question, but, the more and more I saw results like this, the more and more I thought, however it's doing it, it's producing results that are interesting and, at a sort of a higher success rate than a lot of traditional methods of drug discovery. And that was something that was very appealing to me and I wanted to be a part of. So I started talking to Andrew then and sort of ended up as Senior Vice President of R&D here.
So what, walk me through sort of how, that from skepticism to, I want to be part of this. It's, what you did, you said, look, it was showing you
it was the
pathways.
It was the results and the pathway, but also my understanding of AI sort of changed. So, I joined in November of last year, 2019. And in September of 2019, I was reading this article by Bob Temple, who is this senior person at the FDA, and he's been around for a long time. He was involved with the approval of Allegra. And he was talking about how there was this new age in drug development, he called it the age of individualization, or like personalized medicine, right? So recognizing that individual differences in patients contribute to individual variations in response. And he called the previous ages the ages of safety and efficacy, and he was citing regulation. And he said, but for this age, it wasn't really kicked off by regulation. It was kicked off by the discovery of drug interactions with Selding. And I was involved in doing those drug interaction studies, and it was actually those studies that led to the invention of Allegra, which didn't have the problems that led Selding to be withdrawn from the market. But it struck me that what was happening then was like the very beginning of the introduction of signal detection into the pharmaceutical industry. Seldane on average was very safe, so safe that the company wanted it to go OTC. And that's what I was hired in to do, to help them repeat some studies and get it ready for OTC. But then we started getting these reports of drug interactions and arrhythmias, and it was the first sort of application of signal detection in the pharmaceutical industry. Signal detection, signals from individual patients. So looking at individual patients for signals. And at first, the signal detection system was so crude that the only signal that broke through were arrhythmias. And then we learned how to parse a piece of the ECG, the QT interval, as a signal detection system. And, as a result of those drug interactions, FDA also introduced MedWatch, which is a signal detection system to get adverse event data from marketed drugs. And then, that's one of the things FDA, monitors how the drugs are actually working in the marketplace, and are there any unexpected reactions and things like that. And it sort of hit me then that what twoXAR was doing and how they were using AI was actually as a signal detection system, sifting through genomics data and phenotypic data and all these different data sets and examining it with an AI system to look for efficacy signals. And with that sort of reframing of, drug discovery as a problem in signal detection, it makes it a problem that's amenable to a computational solution and allows you to apply, methods of signal detection that have been used in other places, other industries,
right.
To pharmacology. And, the results then it sort of clicked and it fit with the results that I was seeing. Oh yeah, the computer wasn't playing a game to come up with this compound. It was sifting through this data, looking for signals. And then, we tested it, and lo and behold, the signal was validated, at least in the first animal model or in vitro test. And so with that sort of reframing of my understanding of AI, along with sort of defining the drug discovery problem, it started to all fit together. And that's when I thought, wow, if signal detection 20 years ago, 25 years ago, according to Bob Temple, was the thing that ushered in this new era in, development or pharmaceutical development. Well, if we apply signal detection to the beginning part, to efficacy, I think it has tremendous potential. And I'm just surprised it hasn't been done before, because it's really kind of, the results are better, let's say much better than I would have anticipated going into this.
Now, so it wasn't just necessarily the results, but I think if I heard you correctly, it's also this factor of time being shorter.
Yes, it's much more efficient. I mean, twoXAR is a very small company, and yet, they've got 18 programs launched, and I think on 10 of them, we have in vitro or animal pharmacology data with positive results. And, it would have taken years and years, especially for a little company like this, to generate that data with traditional methods. So it's like much, much more efficient. It gets you out of, the triangle that managers are always talking about, cost and time and quality, and you can get any two of those, but you have to sacrifice on the third one. Well, this just sort of, breaks that all open. You can get really good results very fast and, much more efficiently than the traditional approach. So it was just a big jump all the way around from the traditional way of doing things.
Now, the other thing, though, that you said is, and so I'm trying to use these two axes, right? One is time, one is you were seeing patterns or pathways that you were like, not seen that before, and opening up sort of a unique area to look at that you might be able to develop a new molecule against, which of course is always a good place to be.
Yeah, exactly. So that was really the exciting part. So, there's a lot of drug development, and a lot of drugs are like they're, Me Too drugs. They're second generation or third generation of something that does this. And maybe there's incremental improvements, and the third statin that's approved is better than the first one or whatever, but they're working by the same mechanism. What was interesting to me is that, twoXAR's approach and their platform would identify molecules that were potentially effective in a given disease, and the approved ones would show up too, or stuff that might be in development by the companies would show up too, which, gives you some comfort that you're on the right track. But what also show up is stuff that not only that hadn't been tested, but that have mechanisms where there's no drug that's approved or even been tested in patients with that disease that have that mechanism. So it's a whole new way of sort of treating the disease. So it's like the computers come up with a new, or I shouldn't say the computer, the whole system,
yes,
people included, have come up with a like a new hypothesis for what might work to treat a given disease that hasn't been tried before. And then, we test those in animal models, and it would, test 10 of them and 3 of them pop up effective. That was very, very exciting.
Yeah, no, I mean, I've talked to a lot of people where they have it, they have a well understood process of making a molecule, and then they, give it to their system and say, is there a faster way, more efficient way? And the system can sometimes tell you how to get to the same end result in a different way than anybody was classically trained. Right. That might bring down cost and decrease time and so forth. And so this doesn't sound, you can see similarities between all these approaches. Now, the other thing though, that when I talked to Andrew, he's like, his hypothesis was you could shave off 3 to 4 years of time in the whole process. Like, do you agree with that sort of?
Yeah, I mean, from what we've done so far, from the things that we've taken furthest along in and have results back from non-clinical pharmacology models, it was like something like 4 months from saying we want to investigate this disease to having results in laboratory animals, and doing them at CROs, the same CROs that big pharma uses in sort of standard classical models. And most of that time, frankly, was for the actual conduct of the study, not all the stuff that brought us to that prediction. And the stuff that, in a traditional process that would bring you to that prediction might take years. And here, we had collapsed all those initial steps down into one that just generates efficacy predictions that can be, immediately tested. So yeah, there's that sort of time and efficiency aspect of it. And then, when there are new molecules or new mechanisms of action, that made it really, really exciting.
So now, but at some point, the system generates what it suggests, and then a human being, though, has got to look at it and be like,
yes, of course.
Yeah, okay.
Yeah. So I can give you like a flow step, and the first step is like all computers, and then the last step is basically, humans deciding what animal model should we use to test this hypothesis. And then the intermediate steps, span the range of more computer and less human to more and more human. So yeah, after the initial, generation of possibilities, which might be, 1,000 come out or something like that, then there is this winnowing process and ranking process. And the first few steps of that are also computer assisted or AI assisted to rank them or eliminate molecules that might be too toxic or for a given indication, things like that. And there's human intervention along the way until, finally the decision as to which of the molecules to put into a non-clinical model is dependent on human insight and not AI.
So, but at some point, that information, now you are working with the people actually building the models, right? So there's,
yes,
I assume that I think of this as a figure eight, right? It just, at some point there's feedback, there's correction, or there's modification of and then it just keeps going back and forth and just makes it better.
Yes, exactly.
It was interesting because I was talking to someone earlier today from a big pharma and saying, one of the companies I was talking to said they're constantly improving their algorithm. And he says, no, we don't do that at big pharma.
Oh my goodness.
Our model is pretty well set, and we're, unless we think that there's going to be some huge breakthrough.
I think we're on, I don't know. This is a question for Andrew, but version, I don't know, but I'm pretty sure it's in, hundreds of iterations of the software. Yeah. So there's this constant learning process.
So do you see this actually also affecting cost in development?
Well, I think, cost and timing sort of go hand in hand. What really, the way you achieve the timing is because you're eliminating all these, wet lab rate limiting experiments. And those wet lab rate limiting experiments also cost money. So yeah, there's a big cost savings as well as a time saving.
But yeah, I think about it from a rate limiting, but also I think the number of parameters these algorithms can look at is much more than,
Oh yeah,
definitely more than me, right? I mean,
right. This is how it comes up with new stuff that you and I haven't, or other people haven't thought of. It's, I think for one of the programs, Lupus, I think they were like, I was talking to Aaron Doherty, who runs the platform, and it was something like 2.5 billion, pieces of data that were going into this that it was sifting through. So yeah, it's more than humans can handle.
So, and then I always think about like, oh my God, the papers that have to be coming out of this. I mean, at some point, you got to start to let the world know that there's this other potential pathway that you could use this, or just to publish this stuff and say, here's a different way to come at these problems and make it more of a widespread. Now, I know, as a startup, you want to be first and own it all, but I think about that just from a science perspective.
Oh, yeah, I agree. I mean, there is, there's that tension between proprietary and getting the information out. So in terms of results, that's certainly something that we are, publishing. For example, the lupus one, I know that the abstracts have been presented, or, all the meetings are virtual now.
Yeah.
But yeah, that we've presented the data from those studies, and also a couple others like hepatocellular carcinoma. And so the data is starting to come out, and people can, judge for themselves and take a look. But it's very positive.
So now, first, if you were talking to other people like with that have your background, so what, how do you, and I'm sure it's come up I'm sure you had this conversation with colleagues.
Yeah.
What do you say to them, and how do you frame it in a way that they sort of can get their head around it quickly, right? And what are the skeptical comments that you get? Because I'm sure that you and I get probably the exact same comments.
I think it depends upon, it depends upon what they're looking at. So if they're looking at the results of a particular program, if they're looking at our SLE data, I just say, just look at the data and decide for yourself, and then ask yourself, does it really matter, if I came up with the idea or a computer came up with the idea? So there was this game that I used to play at Jazz when we were doing like opportunity assessment, and we called it doing, a pre-mortem. So we pretend, okay, we bought this asset and we did everything and executed according to plan, and it failed. Why did it fail? And if you write down all the reasons it could potentially fail, and the last reason is because a computer came up with the idea, that wouldn't stop us from moving forward with that program, because in the end, the FDA doesn't care if a computer came up with the idea or, whoever. They care about a development plan, about development rationale, and your data. So it's very easy to convince people on individual products. I think on, the AI side, it is more, people are more skeptical, and they're more like in my position. And I have to say, look at, put away your preconceptions about what AI is. And, if you're like me and you thought it was about computers playing games against humans and they win, well, it's a little bit, maybe that's part of it, but it's a little bit more than that. And if you view it as signal detection, that might help you understand what the potential is. And, for me, that helped. And also seeing the results, repeat over and over, convinced me that that was in fact the case. But yeah, we do run into people who are skeptical, say, oh, yep, this data looks really nice, and that piece of data looks really nice, and this program looks good, but they're not, I guess, convinced that there's a block there, and they can't see that, if it works 3 times or 5 times or 10 times, that it's likely to, keep working. And we think that there are 1,000 diseases that it might be applicable for. There's some probably that it's not, like infectious diseases. It's the platform is not set up to do something like that because you've got a third thing, the organism itself. But for other things, it's very broadly applicable.
Yeah, I mean, we've always talked about it, identifying new targets, completely different pathways that have a major effect on the disease that nobody ever considered.
Right.
And then the third is repurposing something that's already out there that might actually have a meaningful effect on this particular disease, but nobody's really using it in that way.
Right. Yeah. Sometimes we come up with stuff like that too. We'll, in our list of things that are predicted to be potentially effective, there might be, a drug that's on the market for something else. So that does happen occasionally.
So if you were coaching somebody and that was working their way through the system, would you tell them to study something? Would you tell them to, read a particular book? How would you sort of coach someone along that was your younger you?
I would say always be guided by science, and look at the results and look at the hypothesis and connect the dots. And if you connect the dots and it says signal detection AI works, trust that and believe it, and put away your preconceptions. You don't have to understand the details of the programming to see the results. And if you can just understand the inputs, and if you can understand the idea that a computer can take 2.5 billion pieces of data and process it, and you can't do that, and comes up with predictions, well, test the predictions. It's sort of empirical. It shouldn't involve, I'm a scientist, it doesn't involve belief or trust or whatever. It's like, here's the input, here's the output. Did the output work? Yes. Here's the input in this experiment, here's the output. Did it work? Yes. How many times do you need to see that to be convinced? And, you can just sort of be agnostic in your beliefs about AI or computers or traditional approaches. And just, just because you're used to doing something one way and it's worked, although maybe, slow and inefficient, don't be closed to the idea that there might be other ways to do things that get you out of that triangle of cost and time and quality.
Yeah, I mean, the other thing that I think about is these systems it's not like they're standing still either. They're in a constant state of improvement and evolution, which is just making them better over time. And we are collecting more data sets that the system can ingest and build into the model. So I think it's moving forward at a pretty,
yes, at a very fast pace. Yeah.
At a very fast pace. I mean, I try to explain the speed at which things are happening to people, and it's very difficult for the human mind to understand. Doubling like has I don't know why we have a hard time getting around exponential growth.
I mean, it's like, science, sometimes things in science move sort of in a linear, predictable pattern, and sometimes it jumps. Like, the cell-vein drug interactions leading to the MedWatch system and signal detection for safety, that didn't come about through a linear process. That was a jump and a response to a problem, and the solution sort of developed de novo, and this is sort of, I think, a similar situation. It represents sort of a break from the traditional way of doing things, and you know, if you're open to an objective to looking at results, you should be sort of okay with that. You know, but there, you know, we get comments from, you know, where's your, you know, 5 KOLs in this disease area who came up with this thing? I said, look, you know, the whole purpose of this, you know, we're a small company, and we've got stuff in that we're developing for SLE. We've got it for oncology indications. We've got some, you know, you can partner with people in various therapeutic areas. We don't have, you know, we hire KOLs as consultants when we need them, but we don't have this, you know, staff of therapeutic disease experts who are, you know, working for 20 years to come up with this molecule. But you know, there's a, you know, to give people credit, there's a lot of companies in the Bay Area that's how they are started. Some professor from Stanford been working on this problem for 20 years, and then they spin it off into a little startup company, and people can, you know, wrap their head around that. Whereas, okay, Andrew and Aaron were working on this computer system, and now we're going to apply it to pharmacology, and we can apply it to oncology, we can apply it to inflammatory diseases, and we can apply it in other areas, and you know, we've got results, and that's just, it's different.
Yeah. I mean, I think, and the other part is if some new pathway comes up that the system came up with, I mean, I'm hard pressed to find a KOL that really, like, you know, would have gone, oh yeah, that was, we absolutely knew about that, right? It's what I find is they're like, oh, I didn't think about that one. And yeah, now that you're showing me the data, that sort of, I can see how that might make sense. It just wasn't right in front of them and obvious.
Yeah, it's what I would call retrospective predictability. It's a sort of that. Now I didn't think of it, but now that the computer did, and oh, that you've got positive results. Yes, I can see how that might work.
We used to call that Monday morning quarterbacking. Like, oh yeah, he should have done that. Yeah, it would have been a better play.
Right.
So here's one of my final questions is, you know, what do you tell those people that say, tell me the first drug that ever got approved by AI? Like, you know, how do you manage that? Because I get that all the time, and I'm like, well, if you wait for that, like, Jesus, like it'll be over.
Right. That's the thing. The product development cycles are so long. If you wait for, if your standard for, you know, accepting a new way of doing something is final FDA approval, you know, you're going to be caught way behind the curve. I think you have to evaluate the compounds as they progress every step of the way and say, you know, what are the results at this step? What are the results at that step? And are they moving forward? Are they moving forward with better success or worse success or the same as the traditional process? I think that's where we're at. I mean, that's one answer. The other answer would be, you know, define what AI is. So, you know, there's AI is sort of, you know, catch-all thing. And, you know, some people would define AI as anything that's more complicated than you could do on an abacus or something, you know, something like that. So,
yeah, I whenever I'm talking about AI, it's a toolkit, right? And depending on what I'm trying to do, I may pull out a different, you know, screwdriver or wrench or whatever, but it's a toolkit of capabilities, processes, approaches that you can take to solve a particular problem.
Right, and people are applying it in other areas of the, you know, drug industry, looking, you know, using AI to develop biomarkers, or using AI even for like patient recruitment, things, you know.
Right.
We're just applying it to the first step in drug discovery.
So how do you think, you know, if you were a betting man, since you were at Jazz working sort of on this stuff, you are somewhat of a betting man because you're betting on something, finding something new. Is you know, big pharma, startups, where do you, you know? I'm sort
Oh,
of betting on startups because I think they're much
yeah,
more
yeah.
nimble and quick. But you know there are good papers in the space coming out from big pharma. It's just I was trying to figure out where do you think the world is
Yeah, I
migrating?
mean, there are certainly, you can find exceptions to the rule, but I think in general, most of the innovation is at small companies. Whether it's, the small companies with the sort of the traditional ones that I mentioned, some professor someplace had an idea that's being commercialized, and then, if it's successful, maybe they take it, or maybe big pharma buys it. That's why there's all these deals. And if you look at
Right.
who's buying what, it's big companies buying the assets of little companies that did the original innovation.
But it's interesting though because I think the buyer set has broadened, right? Whereas normally I would think like it would be okay, Pfizer, Merck, like you go down the list, right? Now I think there's, potentially there's Amazon,
Oh, yeah.
Microsoft, Google. I mean, I was just talking to someone and they were saying, yeah, we keep talking to Amazon about partnering, but they're just missing data. And if they had data, like all of a sudden they become a competitor as opposed to a partner.
Yeah.
So it's an interesting dynamic of new names that are coming onto the forefront.
Yeah, I think my experience, was more with the pharma side, but I don't doubt that at all. That more and more people are getting interested, and maybe the AI component of that is more accessible to people at Amazon and Google than it is to people in the top 5 pharma companies. Maybe they're just more familiar with how to manipulate large amounts of data.
It's interesting because I think you have to have both. I mean, at some point it's producing all this data, but then someone needs to look at it
Yeah,
and think about, okay, okay, that makes sense, right? Or okay, I can believe that. B, how the hell are we going to test that, right?
right.
What's the model and how are we going to, and what's the rest of the process going forward? And that requires some, I wouldn't
Yeah.
want a novice. I'd like
No, no,
somebody who's
no,
been
no.
through that
That's why.
road.
That's why I joined twoXAR to help them with that piece. The program or the platform came up with this, and what was helpful and what sort of distinguishes it is, aside from producing a answer, or quote unquote answer in terms of a prediction, you could trace back why the computer liked this one or why the computer liked that one. And so that was very helpful to me, and that helps you design the appropriate test model to put the molecules in. But a lot of AI, they don't, it doesn't give you the rationale. It's sort of a black box type of prediction,
Right.
and that's much more difficult to deal with.
So this must also spur a lot of IP generation. I mean,
Yeah, absolutely. I think it has the potential to do that. Obviously, I can't talk about.
yeah, no. I'm just saying in general, right?
Yeah, in general, yes.
As you're moving down this road and you're identifying things at a speed much faster than others, right? The IP attorneys must be quite busy, or you hope they would be.
That would be a predicted outcome, yes.
well, Mark, any other thoughts along these lines that you can share with the people that are listening to this? I mean, I have physicians listening to this. I have people in the pharmaceutical industry. I have my wife listens to it, right? As a layperson, it's anything you can share with that group from your experience that you would want them to know.
I would just have an open mind and make your decisions like a scientist based on results. And if you do that, you're on a good path.
Excellent. Well, I really appreciate the time. It was great to talk to you and hear this story. As these products go forward, we may come back to you and ask you to be back on the show.
I'd love to come back. Thank you.
Excellent. Thank you.